start our next chapter. Okay, so we're going to talk about cellular respiration. We're going to talk about how we make energy. All right, so we're going to look at a few things. We're going to talk about how energy is transferred. We're going to talk about the different stages. I think now you know how many stages are there in respiration. There's actually 4. Okay, and we're going to talk about how it's regulated and modified. All right, first thing. Remember we talked about the law of conservation of energy? Anika Armstrong. What does that mean? The law of conservation of energy. You cannot create or destroy what?
Energy.
You can only convert it, transform it from one form to another. Very good. A round of applause, guys. All right, excellent. So you're going to see in respiration, there's a bunch of energy transfers. Okay, you'll see energy transferred in numerous points in respiration, and you're going to see the role of things like electrons over here, oxygen, the proton gradient of hydrogen, and something called substrate level phosphorylation and oxidative phosphorylation. These are all examples of energy transfers that you'll see as we go through respiration. Okay? I haven't explained them yet, so we have to still talk about these. But this is what you're going to be seeing. All right. Now, we are going to look at aerobic respiration. That means we use what? We use oxygen, okay? So just keep this in mind that we are going to be studying aerobic respiration. It's not the only way to make energy, okay? There are other organisms out there that do not use oxygen. They're anaerobes, but we're gonna focus on aerobic respiration. And then later on, we'll talk about, probably not today, talk about fermentation. Okay? So, sugar. That's the molecule we're going to focus on. Trivia question. Is sugar the only thing you can get energy from? No. Can't. There's other things, right? But we're going to focus on the breakdown of glucose. Then we'll talk about how other molecules are used, all right? Now, if you look at this chart, we talked about glucose can react with oxygen to give you carbon dioxide and water. So here's glucose and here's oxygen. If I leave some glucose in a beaker on my desk, if I leave like a little bit of like a clump of glucose and there's oxygen molecules wandering around and bumping into it, nothing happens. What do I need to add to make oxygen bump into glucose with enough energy to get them to react and produce this? Lots of heat. I need to add lots of heat energy. So we don't do this. If you look at what respiration does, it's the same. It looks like it's the same thing. We have glucose, we have oxygen, and we get carbon dioxide and water. But you'll see respiration involves many steps, and we don't need to put in as much energy because we use what special molecules? We use enzymes to run these steps, and you'll see that as we go through respiration. Now, the whole point of respiration is to release what? Energy. But that would be useless if we released it. And by the way, how do you know that this reaction releases energy? Because remember, these are the reactants up here. These are the products down here. Which has more energy? The reactants. So, the products have less energy, which means there's energy being released. What kind of reaction is this then? Review. This is an exergonic reaction. Okay? Now, it's pointless to release energy unless you capture it. So, what's the molecule that captures the energy that we release? What's the molecule that we're trying to make in respiration? It's this one right over here. ATP. Okay? So there's no point in releasing energy unless you're going to capture it. We're going to try to capture it in the form of ATP. But you can see in both diagrams, to get glucose to react and release energy, true or false, we have to add some energy to start. What do you think? Whether it's the combustion of glucose, this is combustion on this side, where we actually get it to react with oxygen using heat, or whether it's cellular respiration on this side, do you see that we have to add energy to even start? Take a look. What do you see here? We're adding what? Energy. What do you see over here? We're adding energy. But what's the difference? With enzymes, you have to add less energy, right? But we still have to add energy. So, what kind of energy do we add? Well, actually, you're seeing it right here. In glycolysis, so you guys looked at glycolysis, you made a flow chart. So here's the picture of glycolysis. Okay? So let's zoom in a little bit. We'll come back to this. These are the many steps of glycolysis. Starting with what molecule? Right over here. This is glucose, right? And then we make a molecule called glucose 6-phosphate. This step here is actually summarized right over here. Okay? So right here. This is the step. Now, this step, does it require energy or does it release energy? What do you think? What do you think is going on here? So look at this molecule right here. Okay? Look at this. Look at this over here. We are converting ATP into what? ADP. What kind of reaction is that? Is that endergonic or is that exergonic? What is this step? It's exergonic, right? Now remember we talked about coupled reactions, which means one reaction does something and it's connected to a reaction that does the opposite. So what's happening over here on this side? Well, this molecule is glucose. This molecule is glucose. What do you notice? What is it? What has been added to it? Phosphate. And it's been added to the 6th carbon. So it's right here. This is the 6th carbon. This is carbon number 6. So from glucose to glucose 6-phosphate, that is an endergonic reaction. Oops. Which means what? This step needs energy. So this step needs energy. This step releases energy. So they're connected together. Okay? Now, look at the name of the enzyme that runs this. Here it is right here. Hexcel Kinase. All right, I'm going to mention something that you should pay attention to, okay? As we go through respiration. If something is a hexagon, how many sides does it have? 6. So the prefix hexo refers to something with 6. So a hexose is a sugar with 6 carbon atoms. So what would be an example of a hexyl sugar? Glucose. What would be another example? Fructose. Okay. So hexyl kinase is an enzyme. How do I know it's an enzyme? Because it has the ASE ending. Okay. And whenever you see the kinase part, it's an enzyme that's involved in adding phosphate. And you'll see some examples when we go through respiration of other enzymes that have the kinase in it. And you probably saw when you built your flowchart with glycolysis or some of the other stages. The kinase tells you that it's an enzyme that adds phosphate. Okay, and you'll see some more examples when we go through this. Okay. So this step here, this is an energy transfer step, but this is energy going into the system. We're not releasing energy, and we're not getting something out. We're actually adding energy to respiration at this point. Okay? You're going to see as we go through respiration that electrons are important. Okay? And you're going to see why. So you're going to see a molecule that is going to come up, and the molecule is called NADH. You may remember this molecule. And this is its reduced form. This is the reduced form called NADH. Okay? It's actually supposed to be NAD+. Actually missing a little plus sign. Okay? So you can see right here, NAD+, that's the oxidized form, and this is the reduced form. And then there's like what I call a cousin molecule called FAD. This is the oxidized form. And then, so here it is right over here, oxidized. This is the oxidized version. And then this is the reduced version. Now, remember what it means to be reduced. What do you pick up? You pick up two things. Electrons and protons. And you're going to see why that's important in respiration. Why do we need to pick up electrons and protons? How does that represent energy? Okay? All right. So, oh, by the way, again, going back to names of enzymes, we're going to learn as we go through this how to decipher what the enzyme does by looking at its name. Okay? So we're going to talk about that. So I'll show you an example right here. So here is an enzyme called glyceraldehyde. It's right over here. Okay? It's called Glyceraldehyde-3-Phosphate Dehydrogenase. Now, we know it's an enzyme because it has the ASE ending. And it tells you what it works on. Now, this is what it makes. Right over here. Bisphosphoglycerate. If you go to your diagram on respiration, this molecule, glyceraldehyde-3-phosphate, is right over here. Okay? It's right over here. And this enzyme is right here, glyceraldehyde-3-phosphate dehydrogenase, and it converts this molecule into this molecule. Now, without even really looking at the molecule itself, you kind of already can tell what happened to that molecule. What happened to it? What did this enzyme do to that molecule called glyceraldehyde-3-phosphate? So, glyceraldehyde-3-phosphate lost something. What do you think it lost? So look at the name of the enzyme. So glyceraldehyde-3-phosphate dehydrogenase worked on glyceraldehyde-3-phosphate. And what did it do to it? Took away what? Hydrogen. So a dehydrogenase enzyme is an enzyme that takes away hydrogen. Okay? So what it did is it actually took away two hydrogen atoms, and it split the hydrogen atoms into two protons and two electrons. The two of the electrons went to NADH, and one proton went to NAD+, to become NADH. So NADH is actually NAD+ after picking up two electrons and one proton. The other proton floats away. We'll talk about what it does later. So, as we go through this, you're going to see names of enzymes, and I want you to be able to decipher what they do based on what their name is, okay? And we'll talk about electrons and protons as we go through this. Why, so like, for example, okay, why do we need to pick up electrons and protons here? Why are they important, okay? And you'll see why as we go through this. Because they're going to represent energy, And you'll see how they represent energy. Okay, so we said that respiration is the process of making energy in the form of what molecule? ATP, right? We're trying to make this molecule right here, ATP. And you're going to see two ways of doing it. I'm going to spend 1 minute on one way, and I'm going to spend hours on the other way. Because guess which one is very simple? The one I'm going to spend a minute on, right? So you're going to see as we go through this, there's two ways to make energy. One is called substrate level phosphorylation, where we make energy directly. So let's take a look at this molecule over here. Okay. This molecule over here, 1,3-bisphosphoglycerate. The 2 means there's 2 of them. Okay, so there's 2 of these guys. There's 2. Okay, this molecule is called 1,3-bisphosphoglycerate. What does bi mean? 2. How many phosphates do you see? There's 1 here, and there's 1 here. This one is attached. So one of them is attached to the first carbon, and one's attached to the third. There's 2 of them, right? Now, look at this molecule over here. This molecule, 1,3-bisphosphoglycerate, becomes this molecule right here, 3-phosphoglycerate. What did it lose? It lost the phosphate on the first carbon. It got removed. You could see it over here. So here is 1,3-bisphosphoglycerate right over here. This is 3-phosphoglycerate. So here it had 2 phosphates. 1 is highlighted. It loses it. Now, if you look at the name of the enzyme that runs this step, what do you see in it? Kinase. Now, remember, kinase refers to an enzyme that adds what? Phosphates. Where did that phosphate get added to, though? Take a look at the picture. Where did it get added to? So what's being made in this reaction? What's going on over here? So it's being added to what? To the ADP. It's being added to it to be converted into ATP. This is a stage where you're getting energy back. Because the whole point of respiration is to make what? Is to make ATP, right? So we're making energy in the form of ATP. Here we're making it. So often textbooks will use this little like starburst symbol to show you energy. So here the energy is being made. Okay? So that's substrate level phosphorylation. It's really simple. All you're doing is taking phosphates from one molecule and literally removing it and putting it onto what molecule? ADP. That's it. That's substrate level phosphorylation. That's all we need to spend time on. Minutes. I said 1 minute. Okay, maybe more than 1 minute. The rest, that's substrate level. We're going to spend virtually the rest of the time talking about how we make it using oxygen. That's what oxidative phosphorylation refers to. Using oxygen to make energy. And you can see it's really important. You're going to see a whole bunch of redox reactions, which involve the transfer of electrons and protons. And the reason why we're going to spend so much time on it is to take a look at this. This is the breakdown of sugar. So you can see we have sugar here, glucose, and we can see the 4 stages. Glycolysis, then this transition over here, then there's Krebs, the 3rd one, and then there's the electron transport chain. On the left and right is what you get from the breakdown of sugar. So you get 2 plus 2, you get 4 molecules of ATP through the direct transfer from phosphate onto ADP through substrate level phosphorylation. 4. But how much do you get using oxygen? That's on the right side. And what do you notice? It's way more. And that's important because what happens if you run out of oxygen? You die. You cannot generate enough energy to sustain yourself. So, we're going to talk about how this works. This is really, really important. Okay, so, what's the organelle that's really important in making energy? Alright, mitochondria, right? Do all the stages happen in the mitochondria? Well, look at the diagram. The answer is, what do you think the answer is? No. What stage does not happen in the mitochondria? Yeah, so stage 1, glycolysis. So glycolysis actually happens outside the mitochondria, in the cytoplasm of the cell. Everything else happens inside the mitochondria. Stage 2, stage 3, and stage 4. What you're going to see as we make energy is a really important process called chemiosmosis. But I'm going to actually come back to that later. Okay? So I'm going to skip over this stuff. I'm going to come back to this later. So let's talk about the stages. So, first stage, glycolysis. Okay? We know, and by the way, this is just the overall summary of glycolysis. Okay? So, let's see if we can figure this out. What is the molecule that we start with? Glucose. Very good. What is the molecule that we end with? This is pyruvate. Okay? This is what we start with, and this is what we end with. Hold on. Let's pause. Okay, sorry. Yeah, so this is glycolysis. We start with glucose over here, and we end up getting pyruvate. What do you get out of glycolysis? How much energy do you get? How many molecules of ATP? 2. And you also make another molecule called NADH through the reduction of NAD+. Okay? So, let me ask you a question. If this molecule is being reduced, remember we talked about a redox reaction is if something gains electrons and protons, so that means something else would what? Yes, I just started recording. So whenever we talk about a redox reaction, if something gains electrons and protons, that means something else must be losing. Okay, so really simple, 2 simple questions. 1, what molecule do we get ATP from? According to this chart, it's glucose, right? Glucose gives you ATP because it becomes, you can see it over here, glucose becomes pyruvate. In the process, you get ATP. But something else happens. If NAD plus is being reduced, that means it's picking up electrons and what? And protons. And remember, it picks up 2 electrons and 1 proton, and 1 proton is just released. Where do the electrons and protons come from? It also has to come from glucose. That means glucose is being what? Oxidized or reduced? Oxidized. So, you'll see why this is important, okay? You'll see why it's important to oxidize the glucose. Why is it important to take away electrons and protons? Now, we know that it's not one step. We know that it's actually how many steps? Many, right? So we know it's many steps.
Glycolysis is the breakdown of glucose into 2 molecules of pyruvate. It begins with an energy input of 2 ATP molecules. First, enzyme action promotes the transfer of a phosphate group from ATP to glucose to form glucose 6-phosphate. The glucose 6-phosphate molecule rearranges to form fructose 6-phosphate. A second phosphate is transferred from another ATP to form fructose 1,6-bisphosphate.
Now, there's a very special enzyme here that runs this step, and if you got it right, you should know what it is. What's the enzyme that makes this molecule? What's the enzyme that makes this molecule? Very special enzyme. And we're going to talk more about it in this, in respiration. I'll give you its first part. It's called phosphofructo. Let's see if we can come up with the ending of the name. Phosphofructo. It's an enzyme that adds phosphates. So phosphofructokinase is the enzyme that runs the step. Now this is an important step because this molecule is highly unstable. That it actually splits in.
This molecule immediately splits to form two molecules of phosphoglyceraldehyde, or PGAL.
Now, just to confuse students, scientists got together one day and said, let's come up with multiple names for the same thing, just to make your lives difficult. No, I'm just kidding. So PGAL is the same as G3P. Okay, so on your flow chart, you have G3P. It's the same as PGAL. All right?
Each PGAL molecule gives up 2 electrons and a hydrogen to NAD to form NADH. Then each PGAL combines with inorganic phosphate to form a molecule of 1,3-bisphosphoglycerate. Each molecule of bisphosphoglycerate donates a phosphate group to ADP to form ATP. With this formation of 2 ATPs, the original ATP debt is paid off. Each of the resulting 3 phosphoglycerates undergoes a rearrangement and then releases a proton and 1 hydroxide ion, which combine to form water. The resulting intermediates, 2 molecules of phosphoenolpyruvate, or PEP, are unstable. Each gives up a phosphate to ADP. Once again, 2 ATPs are formed by substrate-level phosphorylation. The end products of glycolysis are 2 molecules of pyruvate.
Okay, so that's, so you can see it's not one step. It's many steps. Quite complicated. You'll notice at the last step, right over here, I don't know if you can see it. Make it a little bit bigger. So the last step that you saw, you end up with what molecule? Pyruvate. And look at the name of the enzyme that ran that last step. What do you notice in its name? Pyruvate kinase. So, I said kinase is an enzyme that adds phosphate. What did it add phosphate to? Right here. Onto ADP to make ATP. So we can see that we make ATP over here, and we can make ATP over here, and we use up ATP over here, and over here. But if you do the count, you might think, okay, we use up 1, we use up 2, we get back 1, we get back 2. Over here, is that even? Remember one thing. When this molecule split, how many did you get of all of these? You get 2. So you don't make 1 ATP here, you make 2, and you make another 2. So you make 4. And you use up 2. So how many did you get back? Or how many did you make? You made a total of 2. That's why there's 2 over here. Okay? 2 ATP is gained. The second stage is super easy to understand. The second stage, and by the way, where does the second stage occur? Where does it occur? In the mitochondria. So, let's think about a problem here. I like to think about problems. Let's make a prediction. The mitochondria, things have to get in to the mitochondria for them to be used by the mitochondria, right? So, what needs to be in the membrane of the mitochondria for this to work, there has to be two doorways. At least two doorways. This is more than two doorways. You guys play transporters in grade 11 or 10? A transporter is a doorway into a cell, or in this case, would be a doorway into an organelle. So what two doorways must exist to allow substances into the mitochondria? There has to be a doorway for pyruvate, and there has to be another doorway too. What did glycolysis make? It didn't just make pyruvate, it also made NADH. Now, that also has to get into the mitochondria. So the mitochondria is where stage 2 occurs. Okay, so we've left stage 1 now. Now we're going to stage 2. We're going to wrap this up very shortly, okay? So, there are doorways in the mitochondria to allow things like pyruvate and NADH in, because we need those things for the next stages. So let's see what happens in stage 2. So in stage 2, so after glycolysis, we have a stage called pyruvate decarboxylation. Okay? Now, what happens in this stage? Well, we start with a molecule called pyruvate, which is in the mitochondria. So it had to go through the mitochondrial membrane through a transporter, got in, and what happened to it? What do you think these little things are? What are those little things? What kind of atoms are these? Carbon atoms. How many carbon atoms do you see? 3. How many do you see here? 2. What's this then? This is a molecule of carbon dioxide. One of the waste products in cellular respiration is what? Carbon dioxide. What happens to pyruvate? Okay, it gets decarboxylated. So we have decarboxylation. And that makes sense because look at the name of the enzyme that runs this step. What is it called? Pyruvate Decarboxylase. Okay? But what else happens to pyruvate? It also becomes what? What do you make? You make this. And remember, when you make this, that means this molecule is being reduced. So if this molecule is being reduced, what else is happening to pyruvate? Not only is it losing carbon, but it's also being what? It's being oxidized. So pyruvate is also being oxidized. So you'll see again, look, we made NADH. Obviously, it's really important to make NADH. And you'll see why later on, not today, but you'll see why it's very important to make NADH. Otherwise, we wouldn't be seeing it in all of these spots in respiration. Okay? So, the enzyme that runs this step is, where's my mouse? Okay. We say it's called pyruvate decarboxylase. Sometimes when you look it up, it actually has another name, and you can probably predict what the other name is. It's also known as pyruvate, not decarboxylase, but dehydrogenase, because it's removing, what do you think? It's removing hydrogen. So the enzyme that actually runs this step is actually what's known as a multi-enzyme complex. What do you think that means? It's actually made up of more than one enzyme. It does two different things, okay? So one part will do the decarboxylation, removing the carbon. The other part will remove the hydrogen. You'll notice, by the way, this enzyme is inhibited by high levels of NADH, which makes sense because remember we talked about feedback inhibition? When you have enough of something, you want to what? You want to stop it. So what is this step trying to make? NADH. If you have enough of it, we want to shut that down, okay? So, we'll talk about next time, we'll wrap up here. We'll talk next time about stage 3. In stage 3, we need this molecule. This molecule is called acetyl-CoA. The CoA refers to this thing over here, which is called coenzyme A. So let's end up with this. We'll talk about stage 3 next time. What's a coenzyme again? It's a molecule that helps an enzyme or enzymes work. Okay? And we'll talk about this next time. Alright guys? That is it for part 1. That is the end.